So I'll just start, sir.
Please, let's get started.
Good morning, good afternoon, and good evening to one and all present here. I welcome you all to the 12th lecture of our COE lecture series. Today's speaker is Professor Manfred Kappes, a renowned scientist in the field of atomic and molecular clusters. So he completed his BSc in 1977 from Concordia University, Montreal, Canada, and completed his PhD in physical chemistry from MIT, Cambridge, USA in 1981. He became the associate professor in Department of Chemistry, Northwestern University, Evanston, USA from 1989 to 1991. Then from 1991 to till now, he is the professor in Karlsruhe Institute of Technology, Germany. From 2003 to 2005, he was the Dean, Faculty of Chemistry and Biology in Universität Karlsruhe, Germany. He was also appointed as the co-director of Institute for Nanotechnology in KIT, Germany, with Professor Horst Hahn. His research interests include investigation of static and dynamic properties of atomic and molecular clusters, developing ion beam and mass spectrometric techniques, time and angle resolved photoelectron spectroscopy of inorganic and organometallic chromophores. Among many awards, some of the one I'll be mentioning are in 1987, he was awarded Camille and Henry Dreyfus Foundation Award for newly appointed faculty. In 1991, he was awarded Alfred P. Sloan Foundation Research Fellowship, and 2003, John Appleton Lectureship, Brown University, USA. In 2006, JSPS Fellowship, Japan. And 2021, he was appointed as a distinguished professor, IIT Madras, India. He has more than 500 publications with around 26,700 plus citations and a H index of 81. Today's lecture is titled Structure and Dynamics of Isolated Clusters. Sir, we welcome you all to deliver your lecture.
Oh, we lost him again, it seems.
Oh.
Well, he will join. Those of you who joined late, Professor Manfred Kappes was here till a few seconds ago. So we are waiting for him to join back.
He joined again now, sir.
Yeah.
Let's join.
Okay, I'm back, at least with audio. And now you need to let me share my slide again.
Please.
It's not letting me.
Oh.
Otherwise, you might send the PPT to Pradeep and maybe we can upload and you can just get it presented. If some wish.
It's a heavy slide, heavy presentation. Could be. Manfred, is there anyone else in the lab so that their computer you can use?
Sir has joined in another computer as well, sir, I think.
Okay, now you should hear me.
Yes, sir.
And
Yes, everything.
you can see my slides.
Yes.
And I will start. I'm sorry for this. I don't know what is going on. I was in Zooms all day, but anyway. So it's my pleasure to talk to you. I will try and make up the 8 minutes. And what I want to tell you about was a story about superatoms. And to begin with, I just wanted to show the people in India where I am. I'm in Germany. It's a country in Europe which is without Russia, the size of India. I'm 130 kilometers south of Frankfurt. And now I am stuck again. The next slide, okay. So KIT has in Karlsruhe 2 campuses. One is Campus South, where I am at the moment, and North, and the work was done in Physical Chemistry Institute in South and in Nanotechnology Institute in North. And yeah, but before I go into the research, now I really am stuck. Nothing is happening. No slides are moving. Perhaps the thing, okay, now here I am. So the story begins in Bern, Switzerland, and it begins with this paper. On a Saturday morning, I was in the lab taking some mass spectra, and in 1982 of sodium cluster beams with seeded beam expansions, and the sodium cluster size distribution, neutral cluster size distribution, was probed with photoionization mass spectrometry and shows discontinuities. And discontinuities at the time at numbers of atoms which we didn't understand. And 2 years later, or 1.5 years later, this seminal study by Professor Knight and Professor Cohen came out, which provided an explanation for these stability islands in terms of the spherical gellium model, which some of you may have heard about. It's basically like the nuclear shell structure model. It uses a positive background potential, 1 electron per S1 electron atom, and was able to describe for sodium and the other alkalis which were measured at the time, the stability islands, with a very straightforward approach, which basically said we have delocalized electrons and nearly spherical structures. And within a year or two, there were also measurements by the Matsuda group of silver and gold cluster ions, where the magic numbers were also, at the time they were called magic numbers, were also seen and could be assigned to the spherical gellium shell closings at the numbers that you had saw previously. Now with ions, you go up 1. If you have a positive ion, you go down 1 if you have a negative ion. And this was very quickly established, also for copper, excuse me. So the chemist, I'm a chemist, the chemist community in this field wondered very quickly what was the molecular structure of these clusters. And so I spent many years with my group working on trying to determine the gas phase structures of bare metal clusters. So these are
Shrikanta, is there a problem in the lab in network connection?
In my office, not really. So, perhaps in Mr. Kappes' office. I can quickly check.
Yeah, is there something that we can do to help him?
Yeah.
I requested him because he is sitting next to Professor Kappes.
Yes. So he perhaps have some bugs with the computer, so he's restarting and will be joined back soon. Hello?
Yeah, we can hear him.
Yeah. Okay, good.
Those of you who joined late, there is some network issue with Professor Kappes, or it may be a problem with his computer. He is trying to join. He was here till a few minutes ago. He was telling us a very interesting story. So hold on.
So I'm sorry. Now everything is working after the computer was turned on again. And where did I stop?
Well, you were talking about an interesting story of sodium clusters. Yes, and then
Did I get to this picture? Yes.
Yeah, you got to this picture.
Okay. And so the gold clusters structures we were measuring with ion mobility and trapped ion electron diffraction indirectly and more directly, and also by electronic spectroscopy. And in gas phase, structures below 20 atom are charge dependent. And so these are anions. Cations look a little bit different. And at 20, for the anions, there is a tetrahedral structure, which was also found in neutrals by, at that time, Gerard Meyer's group and Lai-Sheng Wang, in one case by infrared multiphoton dissociation, and in the other case by photoelectron spectroscopy. So these are the tools that are used for gas phase structure determination and were used in those days. This is quite a while ago. An interesting thing that it was already known at the time was that the clusters of gold without ligands can form isomers. And in some sizes, there were several isomers coexisting, and it also depended, the coexistence depended on the vibrational temperature at which they were studied. So here you can see an example where 12 atoms of gold as the anion at room temperature can be made into non-interconverting isomeric forms. And this was basically established by ion mobility and also trapped ion electron diffraction. I want to spend a few moments about talking about trapped ion electron diffraction, which is a method that we still use in my group. And here, this is in its present incarnation for bare clusters, but also a method which could be used for the kinds of clusters that are studied by many of the people who are listening to me. So this is a machine in which ions are stored, and then electron beam of 40 kilovolts is sort of transits the stored ion region, and you get a diffraction pattern, which is essentially like a powder pattern. In X-ray crystallography, the ions are not oriented, but they're mass selected. So one can get a size selective electron diffraction pattern on about 10 to the 5 ions in 1 cubic millimeter. So not very many, and limited essentially by the intensity of the cluster source and also by space charging, which is a problem which happens with ions. So in the electron diffraction experiment, the ions are thermalized to some temperature by collisions with helium, and therefore one needs to do background subtracted measurements with and without ions, and one gets essentially these diffraction rings, which are then analyzed with by assuming a model structure, which comes from DFT calculations, and then the degree of agreement between the model and the experiment goes into the confidence within which one can make a statement about the structure of a specific cluster size. So the examples, so this is the machine in the background. We spent many years, for example, looking at 55 atom clusters of the transition elements. But the story today is about magic numbers, so I won't say very much more about that. But the basic message is that for gas phase cluster experiments, particularly on ions, it's a heroic technical effort, and that you need to build machines which are very sensitive, and it as a consequence also is quite time consuming to get such structure determinations. And we have over the years looked at many different elements and have gone from basically an encyclopedic thinking to problem-driven selection of bare cluster systems which are interesting for further study. One thing that's ongoing at the moment in my group is to look at ruthenium cluster anions, which are of interest for ammonia catalysis. And there, the cluster structure as a function of size of the bare cluster is looked at, and also what happens to the cluster structure when it's fully hydrogenated, which can also be done in this experiment by adding, for example, hydrogen or deuterium to the clusters. And this is just meant to be an overview of what goes on. Basically, there's a dramatic structure change when the cluster is hydrogenated, and there are structure type changes, which are important for small clusters and not so important for larger clusters. And there is a relation to what goes on in catalysis on surfaces. But as I'm talking to you, I thought it would be better to talk about some clusters of elements which are of common interest. And there I go back to a picture which I adapted from Pradeep's paper in 2017, which is, I think, a beautiful drawing of the way that the field of ligand stabilized clusters has developed. And for me, originally just observing this from afar, I was, of course, intrigued by the possibility that one can now make atomically precise metal clusters on a gram scale instead of 10 to the 5 per cubic millimeter. And this field started out with Bartlett in terms of the synthesis. So it's driven by synthesis. And then Rob Wetton, who may be listening, I don't know, was involved. And also a little bit before, Matthias Proust and Schifrin in finding ways to make primarily gold clusters. And there's also, I saw Tatsuya Tsukuda, initially at least. He was also with his colleague Negishi involved in making the syntheses more easier. Anyway, so that was the situation in this field going on in parallel to the gas phase stuff. So we're here now, 2003, so 20 years ago. And 2008, it was, I was actually, in that year, I met Hanu at, I think, at the conference, and this was, for me, very interesting observation that at the time, the clusters that were known and their properties, mainly gold, could be explained in terms of density functional theory calculations, which sort of, if you defocused the view of looking at the output, could be explained also in terms of superatoms. So the magic numbers, the spherical gallium shell closings that were already known in the gas phase beam community for the bare clusters. And so the paper, to look at it a little bit more closely, there were essentially, they calculated density of states on the basis of these DFT calculations for various different known clusters, and were able to find P shells and D shells and S shells, depending on the total number of valence electrons. And from a bare cluster point of view, that first of all, one had to try to get one's mind around this. And the way that one at that time, at least, was talking about these was there's a core of gold, then there's a shell of staples, and then there's a ligand termination. And the electron counting takes into account the number of electrons in the core, then some number of electrons which are trapped by the stables, which are then typically subtracted. And then one ends up with counting rules like this for the gold 102, which is then a 58 electron dressed superatom. And this is where Pradeep comes into this story. I said it was a story about atomically precise clusters, superatoms. So Pradeep visited Karlsruhe in 2015 and 2017, and he's also, of course, been here more often since. But this is when I first sort of got into contact with him and with people from his group. And this is where we got into the ligand stabilized cluster field from the point of view of the experiments that we do. And this was a meeting which was also, of course, facilitated by Horst Hahn, who's here in the audience, who helped me to get to know Pradeep. And at the time, Pradeep was working on something which I found fascinating, and his group, that is intercluster reactions. So these clusters that can be made atomically precise, that are now stable at room temperature in solution, and have oftentimes superatom electronic structures, can react with each other in solution by exchanging atoms without the nuclearity of the respective clusters being significantly perturbed. So one ends up by exchanging in the 25 gold cluster golds with silvers, and in the 44 silver cluster silvers with golds. And the observation, I thought, was something that one could perhaps study a little bit further with the methods that are available in my group. Unfortunately, this was possible together with two very talented people who came from Pradeep's group, Ananya Bakshi and Papri Chakraborty, who together with Marco Neumeier looked at the reaction of gold 25 and silver 25 in solution using high-resolution mass spectrometry. And the kinds of experimental data that can be obtained. So this is electrospray ionization, and this is a function of time. The mass spectra that you see are a function of the reaction time in solution. And the interesting thing about these mass spectra is if you start out with the pure clusters, you start not only exchanging the gold and silvers, as I said before, but there's also a significant signal that's observable at a mass to charge ratio corresponding to dimers. So a monomer of gold 25 and silver 25, and later on of mixed of alloy clusters with 25 more enriched on one side and on the other side. And the other thing that's observable is if you take these dimers and fragment them at various times in the reaction, the fragmentation patterns change. Even though the cluster nominally has the same composition, the dimer cluster, the fragmentation patterns change because, as we then proceeded to analyze, in the dimers, there is exchange of atoms, and with time, the composition of the monomers that are exchanging atoms in the dimers changes. And so the process that goes on, we tried to model with kinetics. So we came up with a model which included 600 different rate constants. And basically, the idea is the two monomers come together to make a dimer for a certain short period of time. They exchange an atom, or maybe two, then they go apart again and diffuse around in solution until later on the dimer again is formed. And the process repeats. And the simulations, some of the simulations are shown here on the right. We basically were able to come up with fits to the rate constants and also take into account the diffusion of these large molecules, which are multiply charged in solution, and come up with the story that the what is important in this cluster-cluster chemistry for that system at least, and probably also for the other systems that have been looked at here, is a transient dimer formation, and the cluster falls apart again, and goes on its way and is eventually transferred into gas phase for mass spectrometric detection in the case of our experiment. But the open questions are still here from my point of view. How does the metal atom exchange actually occur in detail? Because the ligands have to move. And then do the alloy cluster monomers that are formed after the dimer comes apart again undergo structural relaxation after they are formed? And this we actually looked at also together with Pradeep using ion mobility. Ion mobility, those of you who don't know, basically the simplest version of this is to take ions through a collision gas cell and measure essentially the time that the ion takes to transit the collision cell. And if there are two different isomers with the same mass to charge ratio, you can get a separation just on the basis of drift time. And drift time can be related to a collision cross section. So the ion mobility can be related to a collision cross section. And then one again has a similar kind of thinking as in the electron diffraction. One needs a structural model and then tries to model the collision cross section. And with some probability, one can describe the observation with a structure that describes the experiment, the experimental observation. Now, ion mobility is a method which we have used quite extensively over the years in my group in other contexts apart from superatoms. And a picture of this technology is shown here as a function of time with regard to the lab in Karlsruhe. We had built many machines ourselves where ion mobility was determined as a function of mass. Then ion mobility was used to separate isomers to look at electronic spectroscopy or photoelectron spectroscopy. And the cluster chemistry, though, we have looked at with a commercial system. Meanwhile, commercial ion mobility spectrometers are to the point that it's very, very difficult for an academic group to actually compete with technology that is commercially available. So the next part of the talk will be looked at with commercial trapped ion mobility spectrometry. And here the ions are, instead of pulling them through a drift cell, they're actually held in place with a radio frequency field, and the gas flows past them. And they're sort of like pearls on a string. They then are separated according to their collision cross-section, and ultimately they are then let out of the ion trap in sequence, and one can again attain a collision cross-section. And this technology we used to study the 25 plus 44 reaction system that I showed you earlier from Pradeep. And Ananya was involved, and Ananya Bakshi was involved in the measurements at KIT. And the question that we're trying to answer here, I already showed you, is after the transient dimer formation and the atom exchange, the atom goes in on the surface of the cluster. Does anything subsequently change in terms of the structure? And the idea is that if the structure changes, the ion mobility will reflect this. And so here, these measurements that you can see are, first of all, mass spectra as a function of reaction time. Now the time is going up in this direction, and you see that with the 44 reacting with the gold 25 with time, gold is incorporated in the 44 atom cluster, which is being probed here. And then after about 12 incorporations, 12 gold atoms, the reaction to first order at least stops. And so this is the sequence of what is going on. And the question is, does this happen? Do the golds from the surface go into the core? And can one look at this experimentally? This core structure is a structure which is known from synthetic work where people have actually gotten a crystal structure of a gold 12 inside a silver 32. But now we're doing this as a probe of solution chemistry and using ion mobility and mass spectrometry to look at the process. And what you see here are a sequence of boxes which show the situation at specific reaction times. And what is plotted is the number of exchanged incorporated gold atoms. So first, there are not few incorporated at short times. At long times, up to 12 are incorporated. And as you can see, lots of things are going on as a function of reaction time. Down here, the x-axis is the new coordinate, which is possible if you do ion mobility. So this is the collision cross-section, which you see is actually also changing. And so collision cross-sections now of specific clusters. Before, we were just showing the entire mass spectra. Now we're looking at specific stoichiometries, specific mass-to-charge ratios, versus collision cross-section and reaction time. There are a number of things that are noteworthy. That first of all, there's collision cross-sections decrease measurably during the course of the reaction. So this is an indicator, for example, of in the gold 10 case, that the collision cross-sections are changing, and there are also multiple isomers which are occurring. And the big picture, the details are not understood, is that first of all, the golds are incorporated on the outside of the silver 44. They replace silvers. Then they move towards the center. And by this process, the collision cross-section is changing. That's measurable, but with ion mobility, or with on the order of it's about at most 1%. No? And so the ion mobility measurement has to be that highly resolved in order to be able to see these processes going on. Now, I wanted to show some additional things. So the message at this stage is the cluster chemistry proceeds by transient dimer formation, and then cluster monomers are formed, which subsequently at room temperature in solution are still undergoing, if you want, isomerization, much like the gold 12 minus in gas phase at room temperature that I showed you before I ultimately went away completely earlier on. And so is there any other way perhaps of getting more information on such processes? And so we have recently, with Papri's help, been looking, or actually Papri's idea, we've been looking at the ion mobility of fragments. So with the hope being that somehow one could also learn about the insides of these clusters, not only their overall collision cross-section. And we go back again to a measurement from the Pradeep's group. At the time, Papri and Ananya were in Pradeep's group. And also about the time that I met Pradeep, they were looking at not only cluster chemistry, but they were also looking at collision-induced dissociation of clusters. And here's an interesting system, a silver 29 cluster with 3 negative charges that when it's looked at by collision-induced dissociation, does a sequential fragmentation in which it loses silver 5 with ligands monoanion in a sequential process. And we decided that we would try to look at this a little bit more closely with ion mobility methods. And this is a study that we have recently done on this system, which is shown Now here again, first of all, on the mass scale, you can see that this sequential dissociation process, in fact, the silver 19 L6 undergoes further fragmentation, perhaps to a neutral species. These are the ion mobility measurements. It was possible to find a way to do trapped ion mobility so as to also study the collision cross sections of the fragments. And we got together with Hannu Häkkinen and his group looked at, with Sami in particular in Hannu Häkkinen's group, and looked at what is going on with molecular dynamics calculations, DFT-based molecular dynamics calculations. And so to describe what's going on, one basically has the constraint of the mass spectra and the collision cross sections, which then are used to screen possible structures which come out of the molecular dynamics. And it turns out that typically stablest forms are compact,3-dimensional, which conform still to the 8-electron shell closing. So the 29 3- is an 8-electron shell closing. The 24 fragment, which is the first that's formed, is an 8-electron shell closing. The 19 is an 8-electron shell closing. And these pictures of density of states are again meant to show that this is to relatively high degree a good descriptor of these significantly, of course, more complicated molecules. So the superatom electronic structure persists even in the fragments. And now, of course, this is with timsTOF. The resolution that is in principle possible on the collision cross sections is about 200. The question arises, can one do these kinds of experiments even better? And now in our group since the beginning of this year, we have the next generation of ion mobility spectrometers, which is a cyclic ion mobility spectrometer. And on the time arrow, so this comes in, was delivered in October 22. And now we are in the middle of using it for many different applications, not all related to atomically precise clusters, but we have also been applying it to atomically precise clusters. Let me tell you a little bit about what this does. So previously, you heard about trapped ion mobility, that the ions are trapped and the collision gas passes by them. In a cyclic IMS, again, the ions are moving through a collision gas, but now the drift cell is not limited in length. It's a cycle. So the ions can cycle around multiple times, and each time the separation between isomers, or the accuracy with which collision cross sections can be determined, gets better and better. So this is the heart. So the cycle. And then there are also possibilities to pre-trap and post-trap, and even to put reagents in, and to put clusters of different charge states and react them with each other. I don't want to tell you about this in any great detail, except that now one can use such a setup to look at, for example, doped clusters, and to look at fragments of clusters. And both of these directions, we are presently in the process of pursuing. Important also, but this is, I think, standard meanwhile, is that the mass resolution has to be high enough to actually see what is going on, even to the level of exchanging hydrogen atoms in such systems. And this is done in the cyclic IMS with a mass resolution of about 100,000. So Pradeep, this is, I was hurrying because I was causing you so many problems. Can I continue a little bit, or should I stop?
I think you should continue for some more time, and we should probably leave about 10 minutes for questions. So maybe 10 more minutes and then 10 minutes for questions.
Yeah, okay. Then what I now would like to do with you is to look a little bit at what the ligands coverage does with regard not only to stabilization, like I said, so the atomic these closed shell systems can be air stabilized by putting ligands on and staples, but also what it does to their dynamics. That was the title of my talk. In fact, part of the title was Dynamics of Clusters. So now I'm going to take you to femtosecond spectroscopy. So we are going to be doing laser spectroscopy on these ions, and this will be done. You've seen basically negatively charged species, multiply negatively charged species. So we are going to be doing laser spectroscopy on isolated multiply negatively charged species. So a way to do this quite well is to do time-resolved photoelectron spectroscopy. So this is just an instrumental schematic. At one end, one sprays the silver 29, for example, and then there's a photoelectron spectrometer and two different laser pulses, one to excite the cluster to below its detachment threshold, and the other one to ionize. And then one measures the kinetic energy of the electrons, which are detached, which are by the ionizing laser, as a function of the delay between the two pulses. And just as an example, before I show you the silver 29, this because it's a system which is very well characterized also in a solution phase. Platinum pyrophosphate, 2 platinum ions that does intersystem crossing in solution from after exciting a singlet state with a lifetime, which depends on the solvent. And in gas phase, one can look at this intersystem crossing in great detail. And after exciting the singlet, the triplet eventually is formed, and then one can detect the triplet because the energy of the photoelectron that is generated by ionizing it is correspondingly different, and then can measure the intersystem crossing rate and can look at other processes which go on. Now, multiply charged, negatively charged ions have oftentimes an additional decay process which involves tunneling of the additional electrons out of the excited states. And this can also show up in the time-resolved spectroscopy. In the case of platinum pyrophosphate, this is a process which happens, and these are some of the rates which are seen. Now, here the interesting thing was the intersystem crossing rate, which in gas phase is 680, occurs on a time scale of about 680 femtoseconds, and intersystem crossing in solution is much slower depending on the solvent. Just to show you how this experiment works. And now we have looked together with Pradeep at the silver 29 3- system, also with time-resolved photoelectron spectroscopy. And so 8-electron system put into gas phase. It has 12 ligands. There are some phosphines which go away when you ionize it from solution, and a structure which is a good description of the species that's probed in gas phase is shown here. And the photoelectron spectroscopy, first of all, one needs to establish what are the electron affinities of the ions in order to understand what's going on. And then the second thing one needs to be aware of when one is studying multiply charged ions is that there's a repulsive, multiply negatively charged ions in this case, is that there's a repulsive Coulomb barrier. That means that if you have photon, which is lower than the top of the repulsive Coulomb barrier in energy, then the electron doesn't come out. And another way of thinking about it is if you have a cluster with 2 negative charges and you want to add an electron to it from the outside, you have to go over a barrier before you end up in the bound trianion. Yeah, so the one photon photoelectron spectroscopy as a function of the detachment wavelength allows you to determine the electron affinity, the third electron affinity in this case, and the repulsive Coulomb barrier. And yeah, so we then used our time-resolved photoelectron spectroscopy to look at the system, similar to how we used it for the platinum pyrophosphate, much bigger molecule now. And this is basically the result of this measurement. And I didn't explain this in detail before, but now I can tell you a little bit more. So what you see plotted is the electron kinetic energy. Yeah, that is the energy that was here in the one photon case. Now we are doing a 2 photon measurement. We're still looking at the electron kinetic energy of the electron that is detected. And this is now a measure of the delay between the first and the second photon, where the first photon does a excitation in the singlet manifold, and then the second photon probes the occupation of the electronic states after a certain period of time. And what can be seen is that there is a significant amount of intersystem crossing in this system to a long-lived triplet. And it's known in condensed phase studies that the luminescence, that there's a weak luminescence which can be quenched with oxygen. So this is consistent with the observation from, in fact, Pradeep's group, that there is a long-lived triplet state which can be formed also here now for the isolated species. And then there are other decay processes which are going on, which are essentially internal conversion. But interestingly, there's no electron tunneling detachment from the excited states here because of the electrostatic barrier, which also has to do, in this case, with the ligand shell itself around the metallic core. And so we believe that this is some kind of charge transfer excitation into in the singlet manifold, in the relaxation of these separated charges leads to, in some cases, to charge recombination induced intersystem crossing. That's the present story. And I think there are many interesting systems or many interesting things of this type which can be looked at by transient spectroscopy in gas phase for such multiply charged Cornish metal clusters with ligands on them. And we are in the process now of studying further examples of these. So I think to keep in time, I should stop. And I want to, first of all, acknowledge the people who collaborated on the work that I showed. That's in particular Pradeep, Ananya Bakshi, who is now at Jadavpur in university, Hannu Häkkinen and Sami Malola, who are at Jyväskylä in Finland, who did the ab initio MD on the silver 29 fragments. And then many people from my group who I want to in particular highlight: Patrick Weiss, our ion mobility expert, Papri Chakraborty, who you've heard about in between, Marco Neumeier, who did the kinetics, Mark Oliver Winghardt, who set up the time-resolved PES experiment, and Aaron Veenstra, who was involved in the studies on the silver 29 time-resolved PES, Detlef Schoos and Dennis Buhmüller, who did the tide measurements, and Horst Hahn, who has been involved with interesting comments and suggestions all along, as well as Andreas Unterweiner, who helped with the femtosecond technology. Okay, that's it. Thank you for listening to me. And again, I apologize for my computer problems. It's all my fault.
Thank you. Very nice talk. Maybe we have 11 minutes or so. I I know that it is a little bit late, but then I know that Tatsuya and Negeshi and people are at home. So maybe you can take this liberty of extending this for 10 more minutes. Others are, well, in India, it's also night, and then they can delay their dinner for 10 minutes or so. So that is why I said that 10 more minutes. So I will ask just one question, which has been sort of troubling me. These molecules are almost like proteins, large systems, and obviously their dynamics, just like protein folding or related kind of problems one can think of, or time scales are of similar kind, could be. So I was wondering, with all these experiments that you presented, what we did as well, what would be the time scale of atom exchange? Can we probe that in greater detail? You are muted, Manfred.
So let me turn off my laser pointer. Yeah. Meanwhile, I'm thinking of an answer to your question. So basically the time scale that we're using mass spectrometry and ion mobility to probe is at best, I think, because of the technologies involved, one could do maybe on 1/10 of a second. Yeah, and the possibilities to change the time that the dimer is actually present together, so to speak, would perhaps be controllable by the viscosity of the solvent. But once they're in the gas phase, then it's then I would say the time scale to probe conformational change depends also a little bit on the resolution with which you want to actually say something about this. Yeah? So the higher the resolution, the longer that you have to basically cycle the ions in the ion mobility spectrometer. So I think it's if you have some way to amplify the structural change, it would be easier. But we're looking, I think, at the moment at things which go on the tens of millisecond time scales. Yeah, if now you mean electronic structure changes, as I showed at the end, that is basically that's 80 femtosecond time resolution, and basically everything longer than that can be done with transient spectroscopy. But to actually have a complete structure determination as a function of time, at the moment with the technologies that there are, I mean, the electron diffraction, for example, is also something which is slow. For ions, yeah, it's been done with large quantities of neutrals with high time resolution, but the electron diffraction, the way we do it, also is probably not feasible on time scales shorter than milliseconds. So I think that one needs probably MD help, and maybe one finds different regions of the dynamical space where one can make experimental statements.
Thank you, Manfred. Let's take other questions. Tatsuya, you had something.
Can I ask a question?
Yeah, go ahead, Tatsuya.
Yeah, thank you very much, Manfred, for the great talk. So, clusters are very flexible in structure and dynamic. And so can you, I hear my voice twice. Anyway, so it's very difficult.
You have 2 machines on. We have 2 machines on.
2 machines are on?
On. One more is there. You are
Okay.
connected to one more. I see another touch here.
Can you hear me?
Yeah, it's fine. It's fine.
Okay. So my question is, can you estimate the barrier height of isomerization in the gas phase? If you control the temperature and
Yes.
Hmm.
So we had published a long time ago a gold 9+, yeah. And that was done with ion mobility cell, which was temperature variable from 100 Kelvin up to above room temperature. And there we were able to see that the two separated isomers as the temperature was increased in between coalesced, and we could get some information on the interconversion rates. Yeah. And from the interconversion rates, one can get some information on the activation energy. And that, in principle, is also possible with ion mobility. Unfortunately, the commercial ion mobility machines that are built for biochemical community, and they are room temperature. Yeah, so it's not with that resolution possible. But in Japan, Professor Misais has temperature variable ion mobility and is in principle at lower resolution. If big structure changes occur, one can get activation energies. Yeah.
Okay. Thank you very much.
All right, colleagues, so there are other questions.
I have a very quick question.
Okay, Sundar.
So this is related to the clusters that we have beautiful clusters with. So is there any like a take-home message that we can probably, like get into a bigger clusters with unknown numbers? It could be with your ruthenium, or it could be gold, silver, or copper. Or is there any strategy? What I would like to know, I mean, there's a strategy to get into.
I think there are different. It's a big field, so there are people who are experts. I think Professor Negishi is listening, perhaps still, in platinum clusters, and ultimately the synthesis aspect is typically synthesis is done in solution, so it becomes a solubility problem. And you have to then find syntheses which are compatible with solubility for larger and larger systems. So I think you're asking me that the atomically precise clusters at what size?
Yeah, well, I mean,
possible?
it's a bigger clusters. I mean, is it like the ligand is playing a big role, or the metal that you are interested in that is playing a role? Or is there anything that like in our chemistry where we have a particular ligand, we know we can reach a particular case size, and after that it basically get fused and like stuff like that. But in your case, like you have very identified clusters, many of those numbers, like magic numbers.
Yes. So I think to answer that, first of all, in gas phase experiments, with laser vaporized particles, you can make clusters with 20,000 atoms, and you can select with a mass filter, if you have enough mass resolution, 20,000 from 20,001. Yeah, but you have a source, a cluster source, a way to make them, which probably makes multiple isomers and is very, very hard on a molecular scale to understand. In solution chemistry, the people who actually are the synthetic experts, I'm not, but the synthetic experts can make, I would say, probably on the order of 500 or so. monodispersed, and maybe Pradeep can comment on that for silver and gold. That eventually runs into problems, the synthetic part, with, I would say, solubility.
Yeah, okay. Yeah, the solubility, of course, it's a big challenge, one of the problems.
Mm-hmm.
Thank you.
All right. Okay. Tatsuya, you have something to comment on, or?
May I ask a question? A small thing.
Go ahead. Yeah.
Yeah. For the multiply charged anion, you detect RCB, repulsive Coulomb barrier. Is this barrier present in the clusters in solution? Barrier is reduced, or barrier still?
The barrier, I think, is it's I think you have to a environment with a dielectric constant, and there's also electron transfer channels, perhaps, which are opened up by the structure of the solvent. And so I think the barrier would change. Typically it will reduce, but it may still be an issue for some chemistry and some solvent. And
maybe
therefore, I would say perhaps the excited state tunneling is something which is also an issue in solution.
Yeah. Maybe it's related to the photostability of such system.
Yeah.
RCB may stabilize system.
Yeah.
Thank you. Thank you very much.
That's a good point. Yes.
Well, we are already at 10 minutes past the deadline. We had a fantastic session. Despite all these hiccups, it was a really enjoyable thing, and lots of open challenges and instrumentation. Probably as we understand systems better with better instrumentation, these flexible systems and their dance, or so their music and all that will become more and more challenging. Greater and greater insights will come. The kind of insights we have just got are about a few atoms, few types of atoms. I wonder what nature has in store when we start expanding this science to other elements, alloys, and a whole range of molecular systems.
May I comment on that, Pradeep? I don't know you then. You wanted to know it, but just a quick comment is that, I mean, one can imagine increasingly higher levels of detail that the experiment accesses, but of course it becomes increasing detail of information. And I think that one probably needs to also to have assistance in experiment using artificial intelligence methods. And this may help then to find systems which are not so easy to describe with theory, where it's important to do experiments. And I think that's a direction which will help to
absolutely. That makes this area, the whole area, very exciting. And maybe with the additional properties of the systems, including many people talk about superconductivity and things. So this system will become exciting. That's all, and let's hear our another colleague in the next meeting. Until then, bye-bye.
Goodbye, and sorry, there were technical problems. Bye.
Bye.
Thank you.
Bye.